antimicrobial. The high saline concentration that's being established can prevent microbes from growing there. Lysozyme, chemical found in our tears, found in our saliva, found in mucous membranes, serves to degrade peptoglycan. So as you can imagine, then it's going to be effective against gram-positive bacteria, more effective against gram-positive bacteria than it might be against gram-negative bacteria. Just as a general statement, and you can of course find exceptions to this, but a lot of the microbes that you find existing on the skin are gram-positive microbes. And a lot of the microbes that you find in the intestinal microbes are gram-negative microbes. So you can see where a lysozyme would be beneficial for tears and saliva and things like that, where the exposure is likely to be to gram-positive microbes.
So it like kind of gives it a little moisture or something?
Mm-hmm. Lactoferrin. You find this also in our saliva and our mucous membranes. Find it in milk. The molecule serves to steal iron away from the microbes. And in doing so, then slows or inhibits the growth of the microbes. One more personalized defense mechanism. Our normal microbiota, the bacteria that we find living on our body and living in our digestive system. These bacteria serve to competitively inhibit the growth of other bacteria. So bacteria on our skin, their simple presence, the fact that they're there occupying space, can prevent other bacteria from landing there and growing. Same within our intestines. The simple fact that we have bacteria there occupying space, they can competitively inhibit or exclude other bacteria. In addition to that, our normal microbiota can use the nutrients that might be in the environment, might be nearby. And so in using those nutrients, prevent other bacteria from becoming established. So they can block binding sites, they consume nutrients. In addition to that, some of them produce toxins that can limit the growth of bacteria, pathogens. A lot of these toxins that they might produce are acidic, and that can inhibit the growth of bacteria. The last thing that we can mention here with regards to our normal microbiota is that they can serve to stimulate our immune system. Their simple presence, anytime you get a cut or a scrape or something, you get introduced to those microbes. Many of us right now are probably suffering from inflammation somewhere on our body. You know, if you've got any hangnails, any little hangnails, any scratches. I got a boo-boo right here. Okay, I'll show you. See, I hit my shin earlier. And if you look at it, you'll see that it's inflamed, it's red. It's because, in just scratching myself, the normal microbiota that I have on my skin were introduced, and this started an immune response, started the inflammation response to kill those bacteria that I was being exposed to. The good thing about that is, when you get exposed to your normal microbiota, they're probably not going to cause you any illness, of course, unless your immune system is suppressed. But it serves to keep your immune system primed and ready to go, so that when you do get exposed to pathogens, your immune system has been somewhat, working regularly. A little bit of exposure now and then to microbes is good for our immune system. It allows it to establish immunity to molecules, antibodies, to molecules that you might get exposed to later on. Questions about first-line defense mechanisms? So before we go any further, let's talk about the cells of the immune system. We think about what their function is. Primary function of the cells of the immune system. Phagocytic. This is not to say that all the cells that we're going to talk about are phagocytes, but one of the main functions of the immune cells are to eat invading bacteria, to remove them from the infected tissues. So they engulf them. They use phagocytosis to do so. Antibodies. We'll see that there are a number, or there are a specific type of immune cell that produces antibodies. These antibodies then attach to antigens and help us then remove those from the system. We'll go more into that when we talk about B cells and T cells in chapter 16. We'll leave it at this for now. We have cytotoxic cells. These cells are not the same as phagocytes. Phagocytes engulf foreign material and debris and dead cells and stuff like that. Cytotoxic cells actually kill our own cells, cells that are abnormal because they're infected with a virus or because they've become cancerous or something like that. So cytotoxic cells are immune cells that actually kill our own cells. And then there are cells that are involved in regulation that are going to amp up the immune system or suppress the immune system depending on what needs to happen. When we think about cells of the immune system and where we're going to find them, well, they're going to be circulating through the blood, but we're also going to find them in the lymphatic system. High concentration of immune cells in the lymphatic system. We'll talk a little bit about the lymphatic system in a minute, but a lot of foreign material and stuff ends up there, and so it makes sense that that's where you'd have a high concentration of these cells. And then we also find immune cells in our tissues. Macrophages specifically will move into tissues and establish residency there. And a lot of times they're the first things that encounter invading bacteria. So let's look at the types of cells specifically. Now your book mentions erythrocytes. I'm not going to say anything about those. Really, those are red blood cells. They're just carrying oxygen, and that's really all we need to say about it. The book also talks about platelets briefly. Platelets are used for blood clotting. Again, that's all we're going to say about them. The ones that we want to talk about are the leukocytes. The leukocytes are also known as the white blood cells. And these are the cells that are involved in immunity. Three types of leukocytes. They're the granulocytes, the mononuclear phagocytes, and the lymphocytes. And we'll look at each of those here specifically. Let's start with the granulocytes. So named because in the early start of this discipline, science, when we were first staining cells and stuff, we noticed that the cells that we call granulocytes, they all had little granules inside the cytoplasm that picked up the stains. The other white blood cells didn't have those granules, and so they were called agranular or agranulocytes. Well, it just happened to be the types of stains we were using were adhering to these granulocytes or granules in the cells. And macrophages and some of the others also have granules in them. They just weren't picking up the stain. So it's a little bit of a misnomer, but the classification continues. The granulocytes called that because they have inside these granules that are going to contain either digestive enzymes or other types of molecules that will trigger a response or cause damage to cells or stuff that's been engulfed. Neutrophils, the most prominent white blood cell, most abundant if you will. About 50% of all white blood cells are neutrophils. These guys are multi-lobed. If you were to look at them under the microscope, what you would see is that they have a nucleus that actually has several lobes to it. And so their nucleus is multi-lobed. If you think about what's going on with these guys, they have granules inside them as well that are digestive, and their main goal is to eat bacteria. They're phagocytic. And so they eat bacteria, and these granules here, stuffed with digestive enzymes, combined with the vesicles that have been phagocytized. We talked about this when we talked about endocytosis. So now you have a vesicle here with bacteria in it. The granules will bind with this, fuse with the vesicle, release the digestive enzymes, break down the bacteria, and this will continue to go as long as the neutrophil is engulfing bacteria. These guys are short-lived though, you know, only a couple of days. They're the first responders to an infection. Basophils. Only about 1% of the white blood cell population. These guys also have little granules, but the granules that they have, instead of having digestive enzymes, contain histamines. And histamines can serve to trigger an immune response. It can serve to trigger inflammation. One of the things that's responsible for the symptoms that you see with allergies, allergic reactions, histamines. Eosinophils. The last of the granulocytes, these guys serve to attack big parasites. Okay, things that are too big to engulf, too big to phagocyze. So,
they serve to attack big parasites.
Big parasites. So imagine a parasitic worm that you might have. Okay, so these guys are going to attack them, attach to the cell membrane, and release enzymes that are going to punch holes into the microbe or the organism, destroy it in that way. Brings us to the mononuclear phagocytes. Make up about 5 to 10% of the white blood cell population. As the name implies, these are phagocytes that have one large nucleus. Separated from the neutrophils with their this is still one nucleus, and we don't want to confuse it with thinking that the neutrophils has more than one nucleus, but the nucleus is multi-lobed, and they're trying to distinguish between that by calling these mononuclear. But recognize all white blood cells really only have one nucleus. And they're phagocytic. Now, when these mononuclear phagocytes are circulating in the blood, we call them monocytes. But monocytes generally are going to eventually make their way into the tissues. especially if there's an infection going on, if they're flowing through the circulatory system, the endothelial cells that are lining the capillaries have special molecules that can grab onto these monocytes, slow them down, and bring them across the endothelial layer into the tissues. When that happens, they're now called macrophages. So once they've moved into the tissues, they are a macrophage. If they're circulating, they're a monocyte. Now macrophages, that's their job basically, professional eater, to go out there and engulf bacteria and debris that they come in contact with. You'll find them in all tissues. And they can, like I said, they establish residency a lot of times, serving not only as scavenger, but also as sentry. One of the first cells maybe that comes in contact with bacteria after a cut or a scrape are the macrophages, because they're in your tissues there.